Okay, everyone, welcome to another expert talk. I just want to clue you in to why I have this little note here. How can we help you? This is from the Pathfinders. You'll see our Pathfinders here tonight, and we're here for you. And we have a first name like McFinn at EverythingALS.org. The same way with Kathleen and John and Joe and Kathy. And John. Hello, John Hudicek. So please tap on a Pathfinder and see what they just might have to offer you, because that's why we're here. And with that,
Welcome, everyone, and thank you for joining today's Everything ALS Expert Talk. We're excited to be joined by Dr. Stephen Kolb. Dr. Kolb is the director of the ALS and Motor Neuron Disease Multidisciplinary Clinic and Research Program at The Ohio State University Wexner Medical Center and a professor of neurology and biological chemistry and pharmacology at Ohio State. His work focuses on understanding the molecular basis of motor neuron diseases and advancing gene based therapies. Today, his team is working on the development and clinical delivery of targeted gene therapies for ALS. We're honored to have him with us and grateful for the opportunity to learn from his experience and research. Thank you for being here with us tonight, Dr. Kolb.
But before I begin, I would like to give a quick shout out to my colleagues at the Ohio State University, and our ALS multidisciplinary clinic, our whole clinical research team, and we've got scientists and students who I have the good fortune to working with. And then also I want to give a quick shout out to my daughters, Zoe and Lucy, who are here in attendance today. Hey, girls. So this evening, I'm going to discuss a topic that it may not invoke the same kind of excitement as a new clinical trial or discovery. But I'm going to talk about an essential ingredient to the recipe in research that leads to those discoveries and that in turn leads to promising new treatments. I'm going to provide a broad overview of ALS model systems, and this might get a bit nerdy. So please interrupt if I'm not clear. And although I suspect that for some of you this might be simplistic, but I hope it's going to be engaging and provide fertile ground for discussions to sprout up. So I have relatively few slides tonight, no data, and this is going to be less of a lecture and more of a kind of a podcast-like conversation. So by the end of this, what I'm hoping is that you'll have a better understanding of how model systems in ALS are made, including the challenges and sort of the important advances that are happening right now to help us stop ALS. And everyone here understands that ALS is characterized by progressive muscular weakness. And we also know that the weakness happens because motor neurons die. And we're going to talk a lot more about motor neurons in a bit. But let's get to the sort of the overall problem that's here. So something causes motor neurons to fail and then die in people who have ALS, even though for the most part, all of the other 85 billion or so brain cells in the brain persist. That's what cell selectivity means. It's just those cells. And so this brings us to this big question of why. Why do motor neurons die? What kills them? And here's the sort of central premise that kind of underlies all of the research that, research labs around the world are doing. And the premise is that if we can understand that, if we can understand the why, then we can stop it, and even better, prevent it from happening in the first place, right? So now there's been much progress toward understanding this question, some of which has been discussed in prior Everything ALS presentations and elsewhere. My laboratory, like many others, is actively pursuing this question every day. But ultimately, and this is the truth, we don't fully understand why. And not only that, the answer to that question may be different in different people. And so then the question that we're going to try to talk about today then is, how do we figure that out? How do we understand how motor neurons die. Does someone have a question or?
No.
Oh, okay. So getting at how do we find out why motor neurons die, the first step is to develop these so-called model systems. And when I have my doctor hat on, I want motor neurons in the people with ALS that we serve to stop dying and, if possible, to repair themselves. That's with my doctor hat on. But with my researcher hat on in the lab, I actually want to kill a motor neuron in exactly the same way that motor neurons die in the body in ALS and then fix it. That would be the perfect experiment. If there was some way that I exactly mimic what was going on in the human body, that would be awesome. But that's what we're going to talk about. That's the tough thing. So let's talk now about what model systems are. We're familiar with the word model for number of contexts. Fashion model wears clothes so that we can kind of see what it would look like on a person rather than just a clump of fabric. A model airplane is a miniature maybe so that you can see how things fit together, and so forth. But a model system that for us is in the disease context is a biological system that's made of living cells that can be studied in the laboratory. And there are mathematical and computational models with increasing complexity. We can talk about those if you want. However, those are also based ultimately on the study of living cells. We can't get around the need to study living cells. So I think it's important to note that some diseases lend themselves to the creation of model systems better than others. And that has to do with sort of the intrinsic nature of those cells. I like to start, kind of with the example of cancer. In cancer, the problem is that normal cells change and lose their ability to control their own growth. Whether it's skin cancer or breast cancer, colon cancer, they all share a common problem of loss of growth control. And as a researcher, you want to find out how to stop these cells from proliferating, stop them from making more. And so in cancer research, you can physically take cancer cells from a person, bring it to the lab, and under the right conditions, those cells will live and keep dividing. It's sort of like gardening, in the lab, you can just feed them and the cells will keep dividing. And there are even cases where you can take the actual tumor, put it in an animal like a mouse, and that mouse will have the cancer. And so if you can find something that kills the tumor in the mouse and cures the mouse, you've essentially made an animal model of that cancer. That's a pretty great step towards actually testing that something to see if it works in humans in a clinical trial. And I like to talk about that because it's in contrast to the biological problem of neurodegeneration that we're faced with here with ALS. Rather than cells that can't stop dividing, we have cells that are dying off and they can't stay alive. And this is not unique to ALS. You mentioned Parkinson's, Alzheimer's, dementia, Huntington's. These are all related as neurodegenerative diseases in the same way that all those different cancers are related, but they have the biological problem of premature and selective cell death. Does that make sense to everybody? Is everybody's on board with that sort of setting the table? Okay. And so, unlike cancers, we can't just scoop out the parts of the brain that we want to understand it from a person. We can't take a spinal cord and look at it without causing a lot of damage. And so that's what brings us to wanting to make these different model systems. That's our dilemma. We want to make a model system before the cells are dying so that we can see why they die and then try to prevent it. So if we want to do that, we need to kind of turn our attention to the motor neuron first before we talk about the actual model systems. And I'm kind of a total nerd when it comes to these cells. I'm obsessed with them. And I'm sort of still filled with sort of a sense of giggles and awe that I can make parts of me move, like wiggling my toes, with the realization that there are brain cells, neurons that are located in my spinal cord that extend all the way out into my limbs and into the muscles in my body, which it seems pretty crazy, when you when you think of it. So here's a quick sort of cell biology and anatomy lesson. And forgive me if I know that most of you are already familiar with this, but I think putting it into context is helpful. So here's just sort of a cartoon, of a motor neuron. Neurons have a cell body there, and there's a nucleus where the DNA is, the genetics is. But neurons also have specialized extensions. You can see around there. I don't know if you can see my cursor or not, but around the cell body that are dendrites. And then there's that long axon that's there, what's kind of short on this cartoon. And that's the output of the cell. And one of the keys about neurons, of course, is that they are electrical. They're electrically active. Electrical signals go in from other cells, from the dendrites, get computated through the cell body, and then there's a firing of a potential across the axon to wherever it's going. And so let me put this up here. So the thing about motor neurons, as you know, there are two types to connect your brain to your muscle. There are upper motor neurons. Those cell bodies live in your cortex up here in the brain, and their axons go uninterrupted all through the brain and through the spinal cord, and go down until they get to the lower motor neuron, which is located in your spinal cord. And this is a cross section of a spinal cord. The lower motor neuron, we're not that clever, so we have upper and lower motor neurons. The lower motor neurons in your spinal cord, their axon is incredibly weird because it actually extends outside of the spinal cord, out into your body, travels within nerves. You can think of nerves as like coaxial cables filled with thousands of these individual axons, and then goes directly into the muscle where it forms this just amazing specialized connection between this axon, this motor neuron axon, and a muscle fiber. This is a neuromuscular junction. Is everybody on? I just want to make sure everybody's on board with all of that as far as what the anatomy kind of thing is. So what's not drawn here is the fact that the motor neurons are surrounded by other cells. So they're not only are they long and go all the way through your body, but there are glial cells that support them, cells like astrocytes, microglia, all these fancy words. The axon that's out there in your nerves are surrounded by other cells. So it's incredibly complex, and it's amazing that it works and that it's functioning. So that's sort of the review there as far as that goes. Now I want to have a little bit. It's fun that you brought up football. I want to have a little bit of audience participation here in a second. So I want us to try to really, if we're going to model this motor neuron, we kind of have to get a sense of what this scale really means. So if you actually measure a spinal cord, a motor neuron in your spinal cord, it's about 50 microns in diameter. That's how they actually are. That's about 0.000005 meters across. It's microscopically small. By neuron standards, it's actually large, but it is obviously small. Now this cartoon is not to scale because the axon, an axon that would extend from, say, the cell that's in your spinal cord all the way out into your arm to your fingers, for example, is as long as your arm and leg. And depending on how tall you are, it could be a meter long or over 3 feet long. And some of these basketball players have motor neurons that are even longer. So you've got a microscopic cell that is macroscopically long. And surrounded by these other cells as I went. So here's a little bit of audience participation here. So I want you to imagine for a second. I'm at Ohio State. Indiana won this year. They were Big Ten, which is great. But we got knocked out. And I want to also give a quick shout out to Bella Therion, who's a recent college graduate who is currently working in my laboratory. And she's going to be entering medical school this fall for putting these graphics together. But I'd like for you to just for fun, imagine that you are holding a football. A football is 11 inches long. And we're going to pretend that that is the cell body of a motor neuron in your spinal cord right now, only we've blown it up to the size of a football. And now I'd like you to imagine that there's this magic string or a cord that you can pull endlessly from the football, and you're going to give the end of that to a friend. And that friend now is we're going to ask them to run the distance of the axon to where the muscle would be if it was to scale. Do you follow what I'm saying? So we're gonna make a huge gigantic motor neuron, but I want to get a sense of the scale. So at that scale, how far do you think the person holding the end of the string, you're holding the football, how far would they have to go in order to get to a muscle, say, that would be in an average person's leg? And for reference, a football field is about 360 feet. Does anybody have any ideas about that? I know you weren't expecting a test tonight, but
I'll say, oh, I was going to guess.
Yeah, Don, what do you think?
100 football fields.
Okay, that's a great guess. Anybody else? Yeah. No, I mean that's pretty long. It turns out it's 51 football fields, about half that. But that's for an average person. And so, who's that kid on the Spurs now who's like 7'4" and like incredibly, so he might have 100 football fields. But just to put that into perspective, if you were holding that football and wanted to throw it the distance all the way to the muscle to be in the scale that your lower motor neurons right now in your body are working on, it would be about that 51 football. And that's 3.5 miles. So that's more than a 5K. You would have to throw that football 3.5 miles to get to the thing. So I get the giggles about stuff like this. I think it's amazing. But the reason that I bring that up is that we are trying to model those cells, and they're freakishly long, and they're freakishly unique with respect to what it is that they do. And I'll remind you that neurons don't divide. So the motor neurons that are making your muscles move right now are the exact same cells that you have had since you were in utero. So, these are robust, incredible, unique types of cells in your body. And that's the thing. Why in ALS do they selectively die? To get back to what our ultimate question is. And that brings us back to this model question that we're looking at. So what is the answer to this question? You guys already have a sense of this, is that to make model systems, there's really two large categories that we're going to review today. And one is cell culture or cell-based model systems, and the other one are animal models. And that's something that we're going to discuss right now as we go. And similar to taking a tumor from a person with cancer, we can't take motor neurons, but we can take other cells from people and bring them to the lab. And that brings us to the first sort of really key advance in ALS research that has, over the last 20 years, really transformed things. And that is what many of you, I'm sure, have heard of, which is this induced pluripotent stem cell, or the advent of being able to create stem cells from adult cells. So in 2006, this was first reported that by two Japanese researchers at Kyoto University, who showed that you could reprogram adult cells and turn them into stem cell-like cells. And they did that in human cells in 2007. And this was a Nobel Prize winning discovery. The fact that you could take a cell that had already decided what it wanted to be when it grows up and turn it back into a stem cell. And the key breakthrough here is that you could take, for example, a skin biopsy and grow fibroblasts or blood cells. And now I can turn those from a person who I see in clinic, I can turn them into stem cells in the lab and then turn them into relevant cell types to study, such as motor neurons or astrocytes and other cells like that. And in fact, that was done not that long after that. So, researchers all over the world created hundreds of recipes where you took these induced stem cells and turned them into other kinds of cells. And in 2008, the first cells like this for motor neurons were derived. And what you're seeing here is a pretty microscope picture of those long axons, although in a culture dish of motor neurons. And you can kind of see the blobs that are the cell bodies. Now, I'll remind you, this is in a small little 7 mm Petri dish. So, this is not the 1 meter long axon that we were just talking about, but nonetheless, it was an incredible breakthrough because it allowed us to grow, so to speak, motor neuron. I'll call them motor neuron-like cells. They express a lot of the things in the lab to study. Does that all make sense to
Yes.
everybody? And not only motor neurons, I should say that once you have that stem cell, you can also turn them into the other cells, like astrocytes and others. And actually, some of my colleagues here at Nationwide Children's Hospital in Columbus and OSU, with skin biopsies donated from our patients here in Ohio State, led to a breakthrough to create astrocytes through this. And so there's a number of these things that are going on, and it's incredibly important. The other virtue of these systems is that they're personal. And one of the things that I know you guys know, and we certainly see this every week in clinic, tomorrow morning is our ALS clinic here at Ohio State, we'll see it again, is the heterogeneity. No one person with ALS has the same rate of progression, the same onset, it is very personal. And with this technology, you can have an individualized model system that's made just from them. And that is where you can imagine developing individualized therapies for specific people. And again, going back to the cancer world, that is the kind of thing that's happening in cancer, where people take tumors, they analyze them, and then they have specialized treatments for them. That's happening in a way now that we have this technology. That's happening going on. Any questions with that? Does that make sense? Cell-based models. And then the second one is the animal-based models. And, I think that hopefully I've already convinced you of how amazing these cells are in your body, but similarly, how deficient cell-based models are to actually be able to mimic the biological task of a motor neuron that is in the body. And that's why we use animals, particularly vertebrate animals that have spinal cords and motor neurons like us. They have long axons, but they also have all those cells that are supported. By and large, depending on the animal, their genetics is quite similar as well, which we'll talk about in a bit. But how do we kill a motor neuron in an animal to understand ALS, like a mouse? How do we start? Like, mice don't have ALS. How do we figure that out? And so let's take a step back and discuss the second key advancement. The first one was being able to make these stem cells from adult cells. The second one is this accelerating understanding and rapid progress in the genetics of ALS. So my colleague, Jennifer Rogenbuck, who is a genetic counselor, researcher, and professor here in neurology at OSU, gave an Everything ALS talk last year where she discussed advancements in ALS genetic testing and counseling and in the genetics, which I recommend that you guys see if you haven't. But for our purposes, I'd like to emphasize that this increasing discovery of new genes, and I'm sorry, this if you can see my cursor, this is number of genes by time, and you can see the rapid identification of all of these are different genes that with different levels of evidence have been associated with ALS. And whereas in 1996 there were 2 genes associated with ALS, there are now over 40 genes linked with ALS. And the reason is because of 2 things. One, because the technology to actually look at DNA has continued to become more sophisticated. But the other big thing is that people with ALS have really showed up and donated their DNA for large research projects like this so that new things can be done. And really, we are humbled by all of the activity that people with ALS do to help with this discovery. So one of the consequences of all of this, new gene discovery is that it brings us to an opportunity to study what the function of that gene is. And again, trying to get to the why. Genes make proteins, proteins do things in cells. And so we want to try to sort of, figure that out in the lab. Here's just another kind of way of looking at that similar data. And this gets back to our third technology that I wanted to talk about. And that is, you've heard of this CRISPR-Cas9 technology, probably, as being this thing. And if you haven't, what it is it's yet another basic science discovery by people who had nothing to do with ALS or disease. That also the discovery of this process won Jennifer Doudna and Emmanuel Charpentier the Nobel Prize in 2020. So these fundamental basic science discoveries open the door because CRISPR-Cas9 allows us to introduce mutations into the DNA of cells and animals. And so, whereas, before this technology existed, we would just maybe find an animal that had something that looked like ALS and we would try to, study that. But now we can engineer mutations that are associated with ALS in humans in mice and other animals much more quickly. And that is one of the things. This is just a fun cartoon, but this gene KIF5A was discovered with specific mutations. And my laboratory took a mouse, used CRISPR-Cas9, and we engineered a mutation for that in a mouse, and are now studying it, just to give you kind of an example of how quickly that can be done. Now, in some cases, mutations that cause disease in humans fail to cause the disease in mice. And sometimes therapies that seem effective in a mouse model for ALS aren't effective in humans. So animal models are not perfect. They're not ideal. And as we learn more from human cells, we are discovering that, some of that may have to do with the genetics of the differences between different animals. But increasingly, the gene discovery and these improving technologies are providing more opportunities for better animal models. This is something we can talk about more if you'd like. So tonight, I really wanted to just provide this perspective on the challenges that researchers face to model these incredible motor neurons that for some reason selectively fail in ALS, but also how these key basic science advances have resulted in tools that are really accelerating our ability and providing much enthusiasm and hope about our continued understanding of the development of actually effective therapies. And hopefully I've gotten you excited about the human-based culture systems that are also increasingly important in the lab, because not only are they important for understanding, but they could also be important in the clinic as diagnostic or prognostic biomarkers. I know you're going to be hearing more about biomarkers on the next talk, but that will wait for another time. So I want to sort of end with two things then. The main takeaway for this really is that is to express gratitude for the generosity of people with ALS to donate their cells and their DNA for research, because this has driven all of those things that I just talked about to develop these model systems. And here at Ohio State, I like to use our Block O thing to kind of talk about the clinical trial, sort of pathway. But we start in the clinic, where you may have genetic characterization, and most people don't have a gene associated with it, but and that's fine. But whether or not you do, by donating fibroblasts or other cells, we can then create these model systems, cell-based models, preclinical models, and they are used to understand the why, and then that can be sort of translated into effective therapies and go back into people with some evidence behind it. And sometimes even just discovering genes can lead directly to those types of things. For those of you who are interested in contributing to the development, and this is just stuff to share, I'd like to give a quick plug for a National Institute of Health initiative called ALS, All ALS Consortium. This is the information. There's a QRS code, but if you Google All ALS, they have a beautiful website there. And they are recruiting people with ALS, but also their caregivers to volunteer and donate cells for exactly this type of work. This doesn't just come to OSU. This is a national repository and goes to more of this development of personalized model systems. And it's an important study that is sponsored by the NIH. There are 35 sites across the entire country. We're one of them in this region. And if you can also enroll remotely if that's it. So with that, I will bring this all to a close. And I really appreciate your patience with me, and I look forward to your questions.
All right. Kathleen, did you have something?
I do. I am curious about this CRISPR Cas19. I hope I have that right. I interviewed one of the reversals, Mark Manchester, when I made a documentary last year. And Mark explained that he felt part of his recovery was the fact that he had an abnormal gene. And this research was probably done some time ago, probably at least 10 years ago. He said they called it CRISPR because it could reach out and ignite other neurons around his dying neurons. So I just wonder if that was a precursor to this new study and new technology on the CRISPR.
That's a tough one without knowing exactly what that is. So the CRISPR-Cas9 technology is actually a very kind of cool discovery that was done in bacteria and other sort of small organisms, and it was just sort of this neat way that DNA could be edited, and then by learning that mechanism, that could be engineered, and we could use that as a tool essentially to introduce things. It sounds different than what he was talking about.
Okay. Well, that was on Healing ALS's site. He has a 3-part series there, but I just remembered that from our time together and just wanted to see if that was on the same track. Thank you.
Of course. Chris.
Come aboard, Chris.
Hi, everyone. So my question is for the doctor. I just wanted to ask if there's anything that he is aware of with the OPCN gene involvement.
So I know that OPCN has been associated with ALS. We also, I'm aware of some families. There's particularly in the Middle East, there are some families that have ALS that seems to be associated with that gene. That's about the extent, though, of my understanding for it. But it is one of those many genes that is being discovered as the technologies and access to DNA testing is increased.
So if you have a particular gene, would that make the CAR T therapy something that is more beneficial than as opposed to someone who doesn't have a gene?
For CAR T therapy for ALS?
Yes.
So I would say no to that. So the genetics, the genetic testing that I'm referring to is really very specific. It's panel testing for genes that we already know are associated with ALS, and this comes from research, but clinically those lists increase over time. Those genes themselves assign risk. They may be interesting. There may be clinical trials and also therapies that may be targeted for them, but those are not genes that are associated with response or not response to CAR T therapy.
Okay, and the donation of the program, do you know how the cells are obtained? Or is it blood? Is it skin
Yeah,
cells? Is it spinal fluid?
yeah. So the all AL for in all ALS, and then on also we for our biorepository, and also throughout the country, different academic clinics have different protocols. But for all ALS, it's primarily blood. Skin fibroblasts can also be collected, and then also CSF is collected as well, cerebrospinal fluid. So really, all of those things can be collected, and you don't have to do all of them. So for you can opt out of things if you'd like.
Okay, and your studies in the lab, is that in a trial yet, or is it?
For the KIF5A work,
Yes, the IPCS
oh, so yes.
cells.
So to be clear, and this is one of the things that I think is why it's so great to have this kind of venue, because one of the things that the use of stem cells, there's therapeutic stem cell studies that go on. So those are where stem cells are used as a potential therapy. And then there's what I've been trying to explain today, which is really using stem cells to create relevant model systems to study and understand and maybe develop therapies from them. So my laboratory is not actively engaged in stem cell therapy. We
Just
are,
studies.
yeah, we're focused more on gene therapies. That's kind of the background for my colleagues and me here in OSU.
Gene
All right,
therapies.
I'm gonna jump in right here. So we're gonna move to our moderated Q&A session. So a
Okay.
bunch of great questions that came in through the chat. If you have a question, put it in the chat. I'm sure I'll make sure we get to it at the end. I just want to make sure that we go through these great ones that came in so far.
Can I interrupt for just a minute? Because I think that Neuron had his hand raised up and he wanted to say something about what was going on. Is that correct, Franciscus? Yes, I'm talking to you, Franciscus. You're on mute, but if you want to say something, say it now.
Are you referring to me? Just checking. Okay. So I'm Francois again, and thank you. I do have a quick question for Dr. Kolb. Is there a way for me to get a hold of you for us to have a separate conversation? That would be great.
Of course. Yes,
Okay.
absolutely. Yeah. I can leave it in the chat here. Maybe that's the best way to do it.
Wonderful. Thank you so much. Very briefly, have you ever been able to witness through neuroplasticity a change actually in the neurodegeneration of the patients or the genes that you're looking at? Have you ever seen any changes from so to speak, neurons that would have been or are in the process of going through neurodegeneration?
That's a tough one. I think that, my short answer would be no, because I think, you're asking a very specific kind of, question. I think that, I would love to be able to induce motor neuron death and sort of see it before it gets, sick and kind of goes through that. I think we do know that neurons that are sick become sort of hyperexcitable. They lose their regulation and so forth. And so their connectivity definitely becomes altered. But again, these are all in model systems, as I was discussing. And what we lack is a good understanding of what's going on in a person with ALS. And that's what I'm also very curious about.
Again, I would love to talk to you.
Yes, that'll be wonderful.
Whatever. Thank you so much.
Thank you.
Bye-bye.
Thank you for allowing me to interrupt.
Thank you, Ivan.
All right, so our first question for you is ALS a disorder where pathogenesis is cell autonomous or cells are being killed by external processes?
Great question. There's no truth with a capital T. I don't know. I think that definitely as ALS is progressing, it's not just the motor neurons that are involved. There is, when a neuron dies, it definitely has an effect on the intimate relationship that all those other cells that I was talking about have. And so therapies that are targeted to other cells other than motor neurons could have an effect on slowing progression or stopping progression of disease. But I don't know. I think that, there are in model systems, there is evidence certainly that the overexpression of some of these mutant proteins that we talk about, if you overexpress them in lots of different cells, that will cause toxicity. But to the extent that that's actually happening in the human, I'm not sure. But so I would say that ALS is probably cell autonomous and has a cell non-autonomous part to it.
Great, thank you. All right, so the next question was from someone who said, we know that iPSC-derived motor neurons are commonly used in the lab, and that TDP-43 can mislocalize from the nucleus into the cytoplasm and axons, where it's associated with increased axonal excitability. There's also evidence suggesting that TDP-43 can propagate to other neurons through synaptic connections in a prion-like fashion. Given that, how do you think about strategies to stop the accumulation of misfolded TDP-43 and prevent its prion-like spread?
Yeah, well, I love that. Those are the kinds of R01 questions that we pose in the lab. So yeah, so whoever posed that is definitely, reading up on their stuff. So, I mean, I think that it would be great. I like to think of dominoes. Something hits the first domino. That's when that first motor neuron, for whatever reason, sort of goes below this threshold and dies. But then there does definitely seem to be this progression of disease that goes in a way that suggests that there is progression from a focal problem into something that is spreading in maybe a prion-like way. And so the analogy would be, if the dominoes are falling, I want to put my hand there so that the dominoes hit that and the dominoes up here keep standing. And so, would there be a therapy where we could try to prevent the progression in cells that have not yet been exposed to that prion? That would be ideal. But I'm hand waving with respect to the exactly how. I think that, the advances, particularly the advances in gene therapy technologies, we already talked about in making model systems genetically. These tools become increasingly advanced, and there are really some really cool ideas going on. I would like to actually demonstrate that progression in a large animal, for example, where you could seed the TDP-43 pathological aggregate somewhere to cause a focal neurodegeneration, and then watch that propagate through. That, those are things that really haven't been done satisfactorily yet, so that we can study those kinds of mechanisms. But awesome question. Of course, I don't have the answer, but I think those types of questions, particularly on the TDP-43 biology, are right on point, and where we're going to be finding some cool things.
Awesome, thank you. So there was some discussion about if iPSC neurons actually are diseased and have ALS, or if they're induced by some sort of change, via virus or heat shock or acid. Do you want to maybe clarify for the audience about kind of using those models?
So, you mean the iPS cells themselves?
Yeah, like if there was some questions on, do they actually have ALS? Are they disease neurons, or are they induced to have disease?
Right. Yeah. No, that's the thing. So one of the, and if I were to show my cards, I prefer the in vivo, the animal models, because of the fact that they have all of the things hooked up. There are real problems, I think, with the induced pluripotent stem cells. There are now, I haven't mentioned, but there's also more direct pathways from, say, a fibroblast into a motor neuron, where you don't go through a full stem cell fate. But you kind of wipe the screen genetically, not with the sequence of the DNA per se, but all the other things that control gene regulation when you make these cells. And the fact of the matter is that in large scales, when people look at IPS motor neurons that other people have looked at and tried to see what's going on there, there's not a lot of degeneration. And not only that, the gene expression from those motor neurons on average is not that different from motor neurons that are grown from people who don't have ALS. And so there are real limitations. I think the utility again comes down to the fact when you're looking at specific questions, and that's really the problem, with those cells. So I think that you can definitely induce them to be dead. I think a lot of people, a lot of labs, there's plenty of ways that you can kill a neuron in the lab. But the big kind of theme that I wanted to try to present, to you is that I want to kill the neuron in exactly the same way that it dies in a human, and I don't think we've done that yet. I don't think we know that yet. It's sort of a catch-22 circular argument.
All right. So the next question, someone said that organoids hold promise to replace mouse models, and neurons on a chip also exist now, although they're very expensive. Do you have any thoughts or comments on these alternative models?
Yeah, no, absolutely. I mean, these brains on a chip, spinal cords on a chip, these all are sort of evolutions of the cell-based models, where you sort of started out with single types of cells in a dish, then you have co-cultures, now you have so-called organoid cultures where you are able to have multiple different cell types in a dish. The sophistication of these things continues to advance, and special culture dishes where you can compartmentalize things. It's all very cool. I think, though, that is all trying to, approximate what's going on so that we have models that we can validate, in the human. So I'm very enthusiastic about the development of the different types of, fancy brain on a chip, motor on a chip. I think, though, that we should not kid ourselves that these are still cell-based model systems that we're trying to optimize and have to mimic as much as we can what's going on in the animals. I also agree that mice in particular have been very unsatisfying in ALS research. And I think that a lot of that has, as we learn more, particularly about TDP-43 biology and the genetics. I have another sort of part of me that is interested in another motor neuron disease, spinal muscular atrophy, where we know that RNA processing of the genome is very important. And so, if you have an animal that has a similar genome, but not similar enough, then you're never going to model the neurodegeneration, in a mouse. And so this, though, leads us to other animals like pigs, for example, that have some of the same kind of RNA processing. And so we here in the Midwest, we have slaughterhouse piglets that we can use as research here. They're treated really wonderful. They're adorable, but we can induce motor neuron loss in them by knocking down a gene that is responsible for this spinal muscular atrophy. And it's a beautiful model for looking at motor neuron death due to that gene knockdown that you don't see as well, or can't study as well, in mice because of their size and genetics. So I think that it's healthy and for a research community for a lot of people to be looking at a lot of different things. And so I don't want to throw shade on one direction or another. I think we're all trying to approximate this amazing, crazy, atypical neuron that we were talking about before.
Awesome, thank you. So next question, something you talked about was individualized therapies. And so if that's possible, in your opinion, how long would it take to get insurance on board? And would advocacy that heads in that direction start now?
Wow. Well, I mean, way premature because we're not, there as far as that goes. I will go, though, into maybe the rare genetic forms. So this is playing out a little bit where there are companies and also philanthropy organizations who are identifying, not just in ALS, but particularly in the pediatric rare genetic disease world, where people are paying for the development of very personalized gene therapies. And so there are many different models for this. It is difficult to get insurers to do this. This is not my thing. In fact, I actively try to not deal with all of that type of thing, and as a researcher. But I think that first, let's get those therapies, and then that stuff, will fall into place. Is my maybe naive view of that.
All right. There are a lot of questions about how people can donate samples, what types of donations, if people could donate outside of the US.
Yeah.
One person in particular said that I have the OPTN gene. So which cells are you asking people to donate and how can they do that?
This is amazing, and thank you so much. So first of all, if you are in the United States, I'd recommend going to all ALS, ALL space ALS. Just Google that. They have a beautiful website, and they can direct you to one of the sites that is near you. And you will talk to a coordinator. If you're in our region, of course, you'll talk with us. But and you can consent remotely even. One of the really cool things about this project is that it is really designed to get people who don't live near an academic center or a big city. And so you can consent and never have to come in and have assessments done remotely. They actually have a phlebotomist, someone who will draw blood, who comes to the house and draws blood. So the first level answer to that is a blood draw. And that's all you would need to get DNA and also cells that potentially could be used for iPS derivation. The second thing would be, if you are at a site like ours, you could donate a skin biopsy or CSF, which is interesting in itself. There are lots of ways, and then of course, some people choose to volunteer for an autopsy after they die, which is really an incredible gift and something that I think sometimes we're afraid to talk about, but it is an important, amazing gift to the research community as well.
Perfect, thank you. So I have an Ohio specific question for you. Someone said, does the state have a Ohio-based ALS registry?
There's no specific Ohio ALS registry. Ohio has, of course, there's an ALS United Ohio group that is amazing here that supports our patients and patients within the state of Ohio. And then the ALS Association also has connections, of course, to clinics that are here. There are really 1, 2, 3, 4, 5 really solid multidisciplinary ALS clinics in Ohio. We are the academic center and sort of the go-to place for a lot of these types of projects, but we work very closely together and are always advocating for one another and for research for our patients.
So someone asked, what exact type of ALS do you work on? Is it only genetic and sporadic? And do you think that sporadic ALS should receive more attention due to its prevalence?
Yeah, I think that this brings up an interesting thing that we've found. When Jennifer, our genetic counselor, started to do genetic testing in all of the patients with ALS, whether they had a family history or not, 10 years ago, this was sort of before this was cool. People were hesitant to get their genetic testing, and if they did get a gene that was associated with ALS, they were often not only were they crushed because they had ALS, but they also had a gene mutation, which also, seemed crushed. We've come full circle now, where now our experience in Ohio is that when we do genetic testing, when most people do not have one of the genes that are associated with ALS, and we find that they're disappointed sometimes because so much attention now is being put on people who have genetic forms of ALS in the form of unique clinical trials that aren't available for people with non-genetic ALS. And I call it, I don't say sporadic anymore. I say non-genetic because it probably all has to do with an interplay between genetics and aging and the environment. But just because you don't have anything to do. So anyway, that was sort of an interesting thing. So I definitely hear and also understand why people with non-genetic might feel like they're getting left out. And so I think that the answer to that, I mean, with respect to my lab, my lab, I don't even really think of the lab, and I think a lot of people like me think this way. I don't think of my lab as an ALS lab per se. I think of it as a motor neuron lab, a motor neuron disease lab. Why, getting to this why do motor neurons selectively die in these diseases? And the reason that we tend to focus on genetic forms, although we have, as I said, worked to develop co-cultures from people cell-based models from people who don't have genetic forms. But the reason that we have focused on the genetic is because as a nerd sort of protein chemist, molecular biologist, it's simple. It's simple to sort of ask the question, okay, this one change in the DNA seems to be important. What does it do? And that's kind of the reductionist way of looking at it, and why the genetic forms seem to be looked at more. But what I would say is that this TDP43 biology and now the genetics of TDP43, that's the link. That's going to be the link between the non-genetic forms and our understanding in genetic forms as well. And that's already starting to happen. So don't feel left out. Researchers aren't neglecting one type of ALS over another. It's more just trying to follow the breadcrumbs towards an answer, as far as that goes.
Right. The next question was, how is Spinraza approved for SMA? And how do they test and approve the drug? Would it be helpful in ALS?
Yeah, no, great question. So that's right up my alley. Spinraza, just for people who don't know, is an antisense oligonucleotide that helps to increase the expression of this one protein that is not expressed enough in babies primarily, although this can affect adults as well, who have this disease, spinal muscular atrophy. And this was, by the way, the leading genetic cause of infant death. So babies would be floppy in their first couple months, and then they would be brought to the pediatrician. They'd be worked up. They'd be diagnosed SMA. They'd do the gene test, and they would basically be told that they're to take your baby home and love them. I actually led a national multi-center natural history study of these babies, and they lived on average 14 months to death. So an incredibly tragic, purely motor neuron, lower motor neuron disease. And Spinraza is one of the drugs that is now a gene therapy that has really been transformative. So that these babies get newborn testing, their gene tests, they can start in Spinraza. There are two other things. There's one other that was developed here, more of a gene therapy, viral delivery. So the question, will it help in ALS? I don't think so. And I think that the reason for that is because people with ALS have plenty of the protein that Spinraza targets. And so I think that I would not waste my effort on that. I would, keep focusing on the why, what's wrong. In ALS, it's not because your SMN levels are low.
Great, thank you. The next question was, there was a lot of talk about heat shock proteins involvement in ALS a couple years ago, but it seemed like it died away. Is there still a place for them in the models of ALS research?
I think so. I think, heat shock proteins are a class of proteins that, they're basically classed that because under stress conditions, their expression is increased. And they have a lot of different actual functions. So it's a heterogeneous group, actually. I think that, it definitely is some evidence that these stress proteins, for lack of a better word, are definitely upregulated, and that knocking them down might help. I think that as far as I know, the clinical trials of medications that went after that pathway have not been successful. And so I think that one of the things in the ALS community that I really think is important is that, once something doesn't work and you're satisfied that the experiment was designed well, you have to move on. You can't keep trying to, prove yourself right. You have to keep moving on. And one of the nice things that's going on, and hopefully I've sort of inspired some of that today, is that there is so much more going on with respect to new targets and more potential therapies that it might be easier now than it was 20 years ago to quickly move on to the next thing and try something else.
Great. The next question was, will patients receive results or findings after they submit their blood sample, and how it's helped?
Yep, this is a great question. So for projects such as All ALS, there was something called Answer ALS before that. There are many others. The answer to that is no, not really. This is research-based stuff. So these first experiments are really more, we're going to grow cells, study them, look at them, and correlate that with that person's clinical presentation and go from there. Now, from the genetic standpoint, right now, well, let me go back. Answer ALS, which was a very similar one, and we were one of those sites. Jennifer and I developed a protocol so that even though people donated their DNA for research, there was a path to receive their genetic results from the research type of thing. And so that right now is being discussed in all ALS. So that is something that may happen. But I think that the way I would look at it is that you're contributing, but not necessarily having a direct benefit from that. What happens then is that when something is learned, or basic principles, then it comes back to you and to other people with ALS to try to help to characterize. So this type of research does not have a clear direct benefit other than the sort of the feeling of contributing to the why.
The next question is, do you look into the role of neuroinflammation in your lab, and what are your thoughts on it as a whole?
Very important. I mean, I think, it's very clear that there is, look, I talked about some of those other cells around motor neurons. No question that there is what you would call local inflammation there. ALS is not sort of a inflammatory disease per se, like multiple sclerosis or other kinds of diseases, but clearly at the cellular level, there is, what you might call neuroinflammation. So I think that's a very good avenue. I don't think it's the only avenue, but I think it's a very important avenue. And I can imagine someday having a neuroinflammatory targeted therapy in ALS along with another type of therapy in combination. My lab, doesn't study that, not because I don't think it's important, but because there's only so much we can study, and I'm not an expert in immunology. I'm more of a biochemist, and so that's why that's kind of how that evolved for me.
Thank you. So this next question is asking for your opinion on what you think drives ALS progression, either an initial change of microglia state or a direct hit on motor neurons themselves.
Yeah, no, I mean, these are some of the $1 million questions, and why we need good, particularly animal models to model progression of disease. I think that, I do definitely believe that there is this sort of nidus, like I said before, along the neuroaxis, somewhere along the spinal cord, that where degeneration begins, and then somehow that spreads. And there's a lot of different, ways. I think through neuronal cells, microglia, for example, with all their extensive extensions through that makes sense. A lot of astrocytes. So I'm open to these things. As a researcher, I'm never going to say, "Oh, that's ridiculous." That's not true. I'm open, but I think that for me, the questions that I'm particularly interested in is the why with respect to the selectivity of the motor neuron itself. And then the other cells, they can all fall into place. But nature's trying to tell us something by giving us all these different gene variants that seem to, result in this selective motor neuron death. I want to understand that.
So this next question from someone who said that they bank their child's placental and embryonic stem cells, and they're asking, would that be beneficial?
I think, I think that that's really cool and really great. I'm not a stem cell biologist. There are people actually whose labs are all about this technology that we talked about. I introduced that breakthrough in Japan, and then subsequently other labs working on the development of these cells. So I'm going to have to punt that one. I think that certainly that's a very unique population of cells that has really more potential to go into cell types, perhaps, or different recipes than the current protocols with adult. But I'm going to punt that one. I just don't know that space.
All right. So next question is, do you look into only motor neurons of the spinal cord and brain, or do you also have research into the neuromuscular junction synapses?
We have we look at neuromuscular junction as part of the motor unit. So the motor neuron, the neuromuscular junction, and the muscle cell itself make up the motor unit. And so on the translational side, or when we are studying animal models that have different types of motor neuron pathologies, we can measure the integrity of that physiologically. So many of you who are online who have ALS have had EMGs, those electrical tests of the nerves. We can do that in animals too, and that way get at the function of the neuromuscular junction. We also, of course, look at it when we do pathology of the mice or of the pigs that we study. So it's definitely a part of what we do. We don't focus exclusively on it, though.
All right, so the last question for you is, are there any ongoing trials that have surpassed a first phase trial that have shown extensive promise?
Just at all.
Yeah.
So yeah, okay. Well, I mean that's a tough one. So of course, you know, as you guys probably know, you know, with my clinical trial hat on, you know, I don't know all of the things because you know, clinical trials that are going on that we're not a part of. Of course, that's confidential until the study is completed. And then once that's completed, then those are announced. And then for the clinical trials that I'm personally a part of with my doctor hat on, I'm not at liberty to talk about because I'm blinded and often to these things, and it's an experiment in humans, and so I don't know you know what it is. So yeah, I wish I mean, I'm not hiding anything from you because I don't know, but I think that you know, the best bet is to, you know, what I tell my patients is, look, as if there's something out there in some trial anywhere in the world that is making a huge difference, it's going to be approved before you know it, and you're going to get it. So don't feel like you're missing out.
So thank you so much, Dr. Kolb. We appreciate your time.
Awesome, guys. Thank you so much, Anusha, and everyone. I really appreciate it. Thanks for having me.
Steve, I got a question for you.
Oh, of course.
What do you have on your finger?
Oh, this is an aura ring. Have you heard of those?
No.
So this is a biometric ring. I have a you know, really good friend who is a triathlete kind of person. And when he and I started training together, he recommended this. But it is it does heart rate and movement and all these things. It's very good for sleep. And so it's so I can see in the morning how I slept based on my heart rate and all these other kinds of things. So I no, this is not an endorsement or a commercial, but it's but I love it. It's a cool little technology because it's data, and I love data.
That's really cool. There's one last question. What's on the desk behind you?
Oh, let's see. What can you see? Yeah. So
Yes.
this is my let me see if I can. This is my my brain, my so is anyone a Young Frankenstein fan? You know, yeah, so this is and then that Steve Martin one where there's the brain in a dish kind of thing. So what my former graduate students who earned their PhD in my laboratory got me this. It's just a fun little experiment mock-up. It's a little plastic brain, you know, in there with a light that's there. So it just a little humor.
Okay.
And actually, Steve, we have one other question for you. What did you think about the questions that were brought to you tonight from our panel, from everyone?
I was very impressed. I mean, you know, I mean, some of them were, as I was saying before, some of them were, you know, right out of a study, an NIH study section. They were very well informed. And I hope that I didn't talk too simplistically about things, but I just wanted to make sure that everybody had, you know, mostly I wanted to share the enthusiasm of the progress that's being made. I think we sometimes get frustrated about the pace of science, but there is real progress that is going on and there are reasons why it's hard. It's a hard problem to solve. So I was extremely impressed by the questions and I really appreciate everyone's attention.
And one thing you mentioned was that we don't really talk that much about, you know, donating our body, but we also don't talk about donating our brain or donating our spine. And these are all very important components of us that will help the ALS community.
Yes, thank you for saying that. And that's absolutely true.
Okay, folks, we had another fantastic presentation right in your living room. Steve, thank you. And to your two daughters, I'm sure they're very proud of you.
Awesome. Yep. Time for bed. So, all right. See you guys. All right.
Thank
Take
you
care.
so much.
Thank you.
All right. Bye-bye.
Thank you.
Bye for now.